All right, welcome back for part 2 of chapter 8, the chromosomes and cell division. We'll get started right away here and finish up before you know it. So we were talking about DNA replication last time, and it's a rather involved situation. First of all, it is directional. The 3' to 5' is the direction that we move, and so we're going from 3' over here. You can look at 3' to 5' in that direction, but it has to be on both sides. And the quandary which a lot of the scientists had was, how is that happening? How can you do both? And one obviously is a nice smooth configuration as it's opening up, being unzipped from 3' to 5, but the other side, that's not so easy. We got to lay down little fragments and things like that. And that turned out to be worked out by a gentleman, and it was named after him, the Okazaki fragments, and were able to do it in piecemeal. It took a while to figure that out, but they did, and it's really very interesting. So the DNA polymerase, you can see at the very beginning, unwinds and separates, it basically unzips, and then you get these DNA polymerase segments here that are literally moving in the pieces as it's needed, the free-flowing nucleotides, and filling in and making those little primers and filling in the gaps, and that's how we do the other fragment. So I actually have a movie that will show that for you. So let's go over it verbally first. DNA replication, the process of DNA replication occurs in 2 steps. We have to unwind it and separate it, unzip it, and then the reconstruction and elongation of a new complementary strand. So we'll go over each of those. Unwinding and separation. Now this is, you can go into damaging the DNA. Obviously you got to unzip it, and you can see which way the arrows go as far as the 3', 5', but it's very much like the analogy with a zipper. In prokaryotes, single origin replication, while in eukaryotes have a multiple origin site, so that each chromosome at the origin of replication is a complex of proteins that bind to the DNA. One of the proteins is an enzyme called DNA helicase that unwinds. It actually makes a nick, causes the DNA to spin around, and then binds it back once it's done doing what it needs to do. Reelongation for the reconstruction part, you can see that obviously as you the DNA polymerase is moving, the lagging strand has to catch up and does it sporadically. So you can see the 2 strands. We have 2 different directions that have to deal with, and I have a movie that will show this, and hopefully we can do this. Hopefully. Let's do the full screen here so everyone can see it. Let me play this video.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome.
Now we have to show this just to show you how well it's
Next,
packed.
the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated.
So now we know how it's packed up. Now let's see how we replicate
Using
that.
computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells.
Look at the complexity.
You are looking
It's
at an
doing
assembly
two
line of
things at the
amazing
same
miniature
time.
biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left.
Isn't
The
that amazing?
whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time.
Isn't that amazing?
The end result is two new DNA molecules.
So, how did you like that? It's an amazing piece of machinery. It's like little fingers threading and making loops, and then the machinery makes the copies. And that's what goes on in 100 trillion cells all times of the day. It's just amazing. And it does it without, what is it, about 99% accuracy, I guess, as they say. And so the enzyme itself is rather complex, as you saw, and it has some rules that it has to follow using a free 3' end in order to extend, and that makes the complement strand here where we have to do the Okazaki fragments problematic. But you saw how that enzyme was able to do that, and it's quite amazing. And so just to remind you that we looked at the unwinding and then the reconstruction of the two processes that we watched. And the 3' DNA polymerase has to have that 3' to move forward, so it's in the 3' to 5' direction, and each nucleotide is added with the DNA polymerase. There are errors, one error per several billion base pairs, which is quite amazing, but on a daily routine, that does introduce some issues, and there are things that can happen during the proofreading. We've already talked about mutations, and we can have sources in our environment. There's just things that we're not even aware of that are just floating around in our environment that can make us sick and cause cancer and various things like that. And of course, just to review, having mismatches as it proceeds, we can incorporate the wrong base, and we get a series of mutations. When errors are introduced in DNA, every now and then we get a new gene as a result, which is part of the evolution. So change is the common factor in these sorts of things. Change is good. The only thing that doesn't change is change itself. And so every time a cell divides, the cell DNA must be duplicated itself into two new daughter cells. The process of DNA duplication is called replication. It's catalyzed by several important enzymes, DNA polymerase and helicase, that has to unwind and do the things that it does. So now let's move on. Mitosis generates replacement. There's two purposes for mitosis: is growth and development. And we know that we have cells that go through various stages. Some cells, as you clean out the intake fan of your computer that you're probably using right now, a lot of that, believe it or not, are dead cells that you see in addition to the hair and junk that's around. A lot of that's just human cells. In animals, the rate of mitosis varies for different types of cells, which is a pretty good idea, because if everything was at the same speed, boy, would we be a mess. Red blood cells replace every 2 to 4 months, and cells lining in the intestines replace every 3 weeks. You can imagine, it's pretty rough stuff. Apoptosis is just a fancy way of saying cell suicide. We have to terminate the cells that become rogue and at the end of their lifespan. So mitosis enables existing cells to generate new genetically identical, and it makes it possible for an organism to grow and replace cells. Now, overview of mitosis. We've been over this in lab. This is a fancy little graphic, but if you look at, zoom to the end, we have 2 daughter cells. You know right there that it was mitosis that did that. This is for somatic or body cells. And if you remember, we go through the various phases to get into the replication mode through that interphase, and we get into mitosis, and we have the various stages of mitosis. And if you recall, we have prophase, metaphase, anaphase, and then eventually splitting with telophase. And pretty straightforward. We've already discussed that. Before mitosis begins, we have to make copies to get ready for the spin-off, so we can create new copies of our DNA. And some of the terminology I just want to make point out. We have the chromosome. We go through replication, and we get 2 sister chromatids formed. We actually duplicate the DNA, and once we duplicate, then we have the sister chromatids, and then they condense more to allow the facilitation of movement for mitosis. So mitosis is the process by which cells duplicate themselves. Mitosis follows chromosome replication and the production of 2 daughter cells from 1 parent cell. So you can see the interphase, and then we go in a nice tight sort of a button, and then as we go into prophase, you see sort of an unpacking, or it is very much larger. You can see the chromosomes. And then we go into metaphase, which they line up in the middle. We've been over that, and lining up in the middle. And the chromatin, by the way, is composed of DNA and proteins that are tightly packed. So we see the DNAs because of all this chromatin fibers that form. And so we see this at metaphase. This is the reason I wanted to kind of step out for a sec. Metaphase, the chromatids congregate in the cell's center. After condensing, the pairs of sister chromatids seem to move aimlessly around, but pulled by the forklifts, or the spindle fibers, the kinetochore that develops in the centromere of each pair, and then they start to pull as needed across. So the spindle fibers are these arrangements that microtubules act as a highway during the cell process, and we have these little kinetochores and these little feet that literally walk along the microtubules, pull. So the microtubules becomes the track, and we pull the portion of that sister chromatid towards the other side, as the one on this side will do that as well. So you can see that we get equal division across. At least that's the intent. Amazing. These are little motors, little kinesians that walk, like I showed you in the class with a FedEx delivery guy. These are essentially the same sort of thing, and they walk along the microtubules to pull the DNA, allow it to separate. Pretty interesting. So then we get into anaphase, as we see now the microtubules and the centromeres pulling we have the forklifts pulling the DNA, those little motor proteins pulling them in as they walk along the plank. The anaphase, you can make that little A, if you recall, and then telophase is at the end, and then we go and divide into 2 daughter cells. So that's the key in mitosis, is that the end result is just two daughter cells. So the ultimate result of mitosis and cytokinesis is the production of two genetically identical daughter cells. Cancer is characterized by an unrestrained cell growth, can cause serious health issues. It's one of the leading causes of death in the United States. Responsible for 20% of all deaths. Occurs with the disruption of DNA interferes, and it just starts to grow wheelie, and it's just not good, and off it goes. They lose their contact inhibition, so they just keep replicating, and we have to deal with that. Now, normally our immune system helps to deal with that. That's why we need to have a good, healthy immune system, but we can't rely on it all the time if we're immunosuppressed. We can see the tumors because during metastasis they actually generate or produce a lot of heat. So we can use a thermal scan and we can actually see where the highest cellular activity is. And sometimes you can find tumors by using a technique like that. To treat it, we use chemotherapy and radiation right now. There are some new techniques that are based on the genetics of the individual. We have better treatments for different types of cancers based on the genetics. But these two usually lose the hair, and but the idea is you get rid of the cancer, and then your body takes over and cleans up the remaining cells, the immune system does, and then deals with it. Cancer is unrestrained cell growth, results from mutations to gene to import. It's important in controlling cell cycle. When those checkpoints are gone, then it forces us to have to deal with chemotherapy or radiation to get rid of it. Sexual reproduction, now this is different. Asexual reproduction occurs when a single parent produces identical offspring. A bacteria will do that. Sexual reproduction produces offspring, a fusion of two reproductive cells, gametes, in a process called fertilization. And we're aware of those things. We've already talked about it. Sexual reproduction in meiosis. So meiosis enables an organism to produce haploid cells. Now that means it's half the genetic content. So we have to have a means to produce these haploid, and so the adult male, it's sperm, and then the adult female, it's the egg, and then allows these two now to do a genetic experiment to reunite the single haploid state to create a new organism or a diploid organism through the process of meiosis we saw up here. And then these cells divide and go through the process of mitosis for the developing baby. So it's quite an amazing scenario. It should be reviewed. The offspring resemble their parents. Meiosis has two important outcomes. It reduces the amount of genetic material. Of course, we need the haploid. And it produces gametes that differ to one another in respect to the combinations of the alleles. And this gives us the variety of offsprings that have unique characteristics. So meiosis is the process by which reproductive cells are produced in sexually reproducing organisms, and it results in gametes that only have half as much of the genetic material, which is haploid, and that allows for the genetic recombination for a full diploid organism. Meiosis only takes place in the gonads, the sexual organ creators, the gonads. Meiosis begins with the cell containing 46 and then ends up, it's essentially mitosis with a bolt-on additional chromosome reduction step. And we see the stages there. So the end of the day, we have 4 daughter cells that they're all haploid. They're all 4 of the haploid. So if we at the end of the day we have 4 cells, then we know it went through meiosis. And so you can see that we've already covered this. So prophase, metaphase, everything is the same as what we're used to. And then we go through prophase again. Really all we're doing is that additional reduction step to get it into the haploid, and then you see we have 4 of the daughter cells being made. Pretty straightforward. And you can look through that slide and make sense of that. Meiosis in animals occurs in gamete-producing cells. It's preceded by DNA replication, 2 rounds of cellular division. The final product of meiosis is in a diploid organism is 4 genetically dissimilar haploid gametes. And I hope that makes sense. If you can wrap your brain around that one sentence, that really puts it all together. And you can see that. Now, sometimes male and female gametes are produced in different ways. The female gamete is rather complicated, and it takes a fair amount. The genetic material, I'm not going to hold you responsible for this part, because it would really take several lectures to talk about how this happens, at least how I recall when I had to sit through this. But the idea is you get a functional gamete, a haploid cell, and then you have polar bodies that are dysfunctional or not functional. And then the males produce a smaller motile gamete, the sperm cell, that billions of them at any one moment in time. And so in species with 2 sexes, including nearly sexually reproducing plant and animal species, females are the sex that produces the larger gamete, and male produces the smaller gamete. Whether it's the male or female gametes that are being produced, the gamete ends up as just 1 copy in each chromosome. And that's obvious, we already know about that. We do get the crossing over event, and when they cross over they make an X, and that's what they call a chiasmata. It's the point where they do cross over, and what they do is they swap the positions where the genes are for the various alleles, and you can see we get a little bit of blending effect in the offspring as a result of the crossing over, and that's the key. Although it doesn't create a new version of any alleles, crossing over just sort of disperses them in a different sort of varied configuration, so it makes it interesting as to what the offspring look like. And you can see with all the different color kittens and various aspects. As a result of crossing over, we believe that the hair code of these cats is rather complicated in terms of the genetics that go on, and it's quite interesting if you're interested in those sorts of things. Of course, I went through a lot of that when I was in school. Sexual versus asexual reproduction. Bacteria produce asexually, where most plants and animals produce sexually. And there's reasons for both. Bacteria, they have to grow and deal in their environments very quickly. 2 fundamentally different ways that cells and organisms, mitosis and asexual, and meiosis and sexual reproduction. And different animals have more complex life cycles and stuff. It can get really challenging, and lucky ours is pretty straightforward. It leads to genetically identical offspring that carry all the genes that the parent carried, which is a good thing. Sex is determined in humans by the last pair of the chromosomes, the 23rd pair. X and a Y is a male, and females are XX. And you can see the little blue Y chromosome and then the larger X chromosome. So determining sex is really dependent on fertilization, XX or XY, and it's really that simple the way they fall. There are some extraneous things that can affect that. We talked about temperature and that sort of thing. Hermaphrodites produce both male and female gametes. I had a dog that was in a puppy mill that was given to me. I don't usually get those kind of dogs, but anyhow, a puppy mill dog. My wife gave it to me, and one of my friends is a vet, and I don't practice, and so he gave me a bill for both spaying and neutering, and what's going on, and he said the dog that I gave him to spay was actually a hermaphrodite, and he didn't charge me for both, but I was quite shocked to learn that it was a Sheltie I had. It was a nice little dog. Birds, females determine the sex of the offspring. Ants, bees, wasps are determined by the number of chromosomes sets that each individual has. It's quite interesting. And turtles, the environment, the temperature of the environment determines the sex of the offspring. It's quite interesting. So there's a variety of methods for sex determination across the world of plants and animals. In humans, the sex chromosomes carry information that directs a growing fetus to develop as either as a male or a female. It's usually determined in the first 14, 15 weeks. As a consequence, sex determination depends on the sex chromosome inherited from the father. Can the environment determine the sex of turtle offspring? Well, yes. I had already mentioned that in warmer temperatures, most eggs hatch as females. In cooler temperatures, most eggs hatch as males. And it's all about, if you recall our discussion about the G and the GC content, A's and T's and G's and C's. ATs only have 2 of the hydrogen bonds, where G's and C's have 3. So my guess is this temperature is related to that. So they were just proving that they had differences in temperatures, and the incubation temperatures, if you have a control, would prove that. So you can read through that if you're interested. Observations of some lizards and turtle species reveal that sex determination is influenced by temperature. Both the lab and natural habitats at cooler temperatures, more males develop. So it leads to the idea that if we have climate change, getting warmer or whatever, it's going to sway the population. Now, we also talked about in lab Down syndrome and having changes in the is evidenced by the karyotype, and having a trisomic 21. If you have a normal set, you'd have one additional chromosome at 23 for Down syndrome. Now we can use methods to check for the presence of any type of condition. Usually it might show up in the urinalysis, might be strange, or blood work, and then the doctor will ask for amniocentesis, which you take a needle, and you can see the visualization equipment here to make sure the needle, when they take a sample, isn't going to hurt the baby in any way. There is risk, but it's pretty low. And then based on that, a karyotype will see, oh, there's 3, and then I'll make decisions as to the parents prepare for raising a child with Downs. Non-disjunction is usually the cause of this. It's where they're supposed to be attached, and for some reason that the tubule didn't form or whatever, and it didn't get pulled properly. So one of the chromosomes remained in the cell, so it didn't separate properly. Karyotypes are visual display of a complete set of chromosomes. A karyotype is a useful diagnostic tool because it can prepare in fetal development to assess whether there's an abnormality number. So you can find those by using a karyotype and having a karyotypist do the work. And there's other ways to do those now with genetic sequencing and becoming really popular. It's usually fatal to have too many or too few of the non-sex chromosomes. And you can see that here's a monosomy Turner syndrome, and where you have only the X chromosome and you're missing the other. 1 in 5,000 carry the X, and you're calling this Turner syndrome, denoted as X. And these individuals only have 45 chromosomes, so that gives a series of changes that we can see summarized here. And it's just interesting. It's dose effect and having a slight difference X and Y. So the Turners have just the X, and Klinefelter's is XXY, Klinefelter syndrome male. And you can see the listing of some of the changes there. So it happens in 1 in 1,000 males have the genotype the XXY, and what they call this a Klinefelter. And metafemales, we can see XYY male and an XXX female. We don't have a name, official name for the XXY. It does occur 1 in 1,000 sometimes, and The other is the super females, it's about 1 in 1,000, and we just don't hear much about it, but it does cause some changes in here's the Klinefelters and some of the others, but it's fatal to have one or too many or too few of the sex chromosomes. Individuals born with a single sex chromosome, that is an X or an XY chromosome, usually survive, but they do have issues. So that's the end of this chapter. Again, this was loaded with vocabulary. Please study the vocabulary. That'll be a big function. I'm figuring out how we're going to do the exam 3 for this portion. I was thinking maybe 2 quizzes, or I'll get to you on that. But I will be sending around a take-home exam for this section. So please keep up with those polls. That helps me do the attendance, and also gives me feedback as to if you're liking or doing things. Again, the teams are strictly for you to interact with other students if you want to work together on various things. Teams works out really, really well, but I'm not making it mandatory. I know that there's enough change that you have to ask for too much. I do have my office hours on Zoom. It's really easy. Just click the link and boom, there I am from 9 to 10 and from 1 to 2 Tuesday through Thursday. And if you have any gripes or questions, or you just want to say hi, that'd be great. Love to see you. And please wash your hands off and don't touch your face. Limit any close contact with anyone. Do not go outside until instructed to do different. It's just not worth the risk until this infectious period is over. Take care.